Good afternoon. My name is Randall Kay. I'm the Chief Medical Officer at Longboard Pharmaceuticals. I'm also a first-time LGS Foundation Family and Conference participant. And I can tell you, thank you. What an inspiring couple of days. It's so nice to start to be part of this community and get to learn, and very appreciative. I had lunch and dinners with different people, the way they've been able to share information with me. It's super valuable. I've been involved for drug development for about 30 years, and I know I don't look that old, but I am. But when I sort of reflect back, I was originally trained as a pediatrician, and I can still remember to this day that first child that I saw that was ultimately diagnosed with a seizure disorder. In this instance, it was infantile spasms. But back then, let's just say the late 80s, maybe early 90s, it was a very different time in drug development. There was a lack of something that we've talked a lot about over the last few days, and that was hope. We had very limited medications that were available to us, very limited options, and there wasn't a lot we could really talk to families about. And then having been involved in drug development over the last 30 years, something remarkable has changed, and something that's me really excited about being here today. I'm going to tell you a little bit about the medication that we're working on, and talk to you a little bit about the study that we're involved in. So I'm going to go a little sciencey. I kind of find this stuff sort of cool, and I hope you'll find it rather interesting as well. So I'm from the company that has a product without a name. Larry, I think we were talking earlier that maybe you can help me figure out the name, but we'll figure that out in time.
Small commission.
Yes, small, yeah. So LP352, as we work in drug development, we sort of think of ways of targeting problems, and we take by precedent knowledge things that we happen to know. So our purported mechanism of action is we are targeting a specific receptor, 5-HT2C. And there's a little bit that we know about that particular receptor, is that we know GABAergic neurons suppress hyperexcitability, or will create an eventual signal that can precipitate a seizure. But there's some other information that we know. If you take a mouse and you knock out that receptor and you just totally remove it, these mice have spontaneous seizures and also decreases their threshold. We also know there's a genetic model, a really fascinating model. It uses a zebrafish, kind of common fish that we like to put in our aquariums. And it's a model of Dravet syndrome, where a 5-HT2C agonist actually decreases the seizure-like activity that these zebrafish exhibit. Now, when I mentioned 5-HT2C, you heard of one of these medications earlier, which is Fintepla, and another one called lacostrine that interacts with that receptor. So here's the approach that we're taking. We think of it in terms of targeted selection. We know that you can target a specific receptor and try to see if there's a way of not targeting the ones that don't. So if you think of the endogenous chemical, a chemical the body makes called serotonin, it can interact with all sorts of these receptors, A, B, and C. But we've learned C appears to be the one that's helping most with efficacy. And if you interact with A or B, it could be contributing to adverse events. So the approach that we took is, well, let's make a compound that's really, really potent at C. And we call it a superagonist because it's even more potent than the endogenous compound serotonin. And then we've, through clever chemistry, found ways to actually not interact at all or negligibly with A and B. So the promise of the medication, and we have years to go in terms of drug development, but the promise is that potentially a more efficacious medication with a great safety profile. So now we have to go into the clinic. So we move forward, and we tested our compound with this zebrafish model. And it's literally these fish are swimming in an aquarium. You add your medication into the aquarium. You would drop food in, and then you just measure these epileptiform events or activity. So to not make it all that complex, the bar that you see in the middle of each of the graphs, the big green one, just shows a high degree of this epileptiform events and activity. And then on the right in blue, you see a marked reduction by adding in the medication that we're developing, LP352. But this is in fish, so obviously we want to see if it might actually work in humans. So we ran one particular study in our phase 1 program to look at a marker. We know if you interact with this receptor in a human, you should see a chemical called prolactin increase. So what we found is, as we increase the amount of our compound, LP352, in the lower doses, not much activity, and then as we got into these sort of medium numbers, we started to see more activity, and then it sort of plateaus off. So when you get to that kind of orange and red bar, you see a sort of plateauing. So it gives us some hints when we go into the clinic. It gives us some hints on what is the right dose to be considering. I have no idea why we call our pharmacokinetic studies SAD-MAD, because it sort of implies something sort of weird and unhappy. But SAD stands for single ascending dose, MAD is multiple ascending dose. They're actually really important studies that are done early in development, and we learn important things. We learn that this medication is rapidly absorbed. We learn a really important thing, especially for your loved ones, potentially, is no appreciable food effect. So it gives us some hints on that we won't have to pay attention much to whether someone has food on board or not. And then we learn a number of other parameters. Now, all the information that we learn from these SAD-MAD studies and overall safety just gives us one major approach is how do we enter into what's important is to start the pivotal clinical trials to understand the safety and efficacy of our medication. So I'm going to talk with just one slide about the clinical trial that we're involved in. It started a few months ago. We're in the very early stages of this study, but just one comment first. I can't tell you, either as a clinician or a drug developer, whether this is going to be the right trial for your loved one. I can't even tell you whether you should be involved in this particular trial. But having been involved in drug development for so many years, the only way we could discover and develop new medications is for people to get involved in research. So a clinical trial might not be the kind of research that you want to be involved in. Maybe it's some of the surveys that we heard about before, but this is really critically important. And I think someday in diseases such as Lennox-Gastaut, the shift will gradually be that research is part of the treatment paradigm in the way it is with a couple of other devastating conditions that families are dealing with. So the Pacific trial design is a phase 1b/2a study in developmental and epileptic encephalopathies. What sets this study apart from many of the other clinical trials, some of which you've heard today, is it's not just in patients with Dravet or Lennox-Gastaut or tuberous sclerosis. It actually takes a wide variety of patients that have these developmental encephalopathies. Sorry, this is a hard word, in terms of the DEs. And so rather than just focusing on one, we're allowing for a patient to have any of the underlying 25 or 26 DEs. It's a 50-patient study, 40 patients on LP352, 10 on placebo. You start with randomized to one of the two treatment groups, so more likely that you would be on medication than placebo. There's a couple, 2 weeks of dose titration up and down based on tolerability. At the end of that time period, the subject patient is locked in at that dose for 60 days in the maintenance period, and then begins the down titration. Currently, the basic information is about 25 sites across the U.S. That number will likely go up a little bit higher. What you're seeing here is an age group of 18 to 65. We are expecting to lower it into the adolescent population, so down to the age of 12 shortly. But as I mentioned, we're in the very, very early stages of the study. I believe we have 4 activated sites in the U.S. and are expecting more over the course of the next couple of months. In terms of the DEs, we described the types of background, typically looking for patients with a minimum of 4 countable seizures per month. historically over the last 2 months prior to screening. On the right-hand side, we're describing some of the key efficacy signals. What we're looking for in this type of a study, the predominant focus is on safety and tolerability, and the pharmacokinetics of our medication. But because of the size of the study and because we are specifically looking at patients with a variety of underlying DEs, we will be able to look at the potential for seizure frequency reduction. We'll also be able to look at their different responses based on the underlying DE or the phenotype, the type of seizures. Is there a difference between a patient with tonic-clonic seizures, as an example, compared to some of the other phenotypes? The one key exclusion criteria I just want to mention, no use of fenfluramine within the last 30 days, same as lacostrine, which I think is available as a named patient program. But as long as have not been on it for the last 30 days, that still is acceptable. So we're excited about this study. My colleague Dewey, who is in the audience somewhere, we do have some brochures. If you are interested in getting some background information, we'd be more than happy to provide that for you. And Tracy, I don't know if there's time for one question.
Yes, definitely. You have time for probably one or two. Any questions about the study? Just put your hands up. See if any questions at home right here.
The question is there an extension? We are planning an open label extension. We're discussing it with the FDA, but we would be expecting that there would absolutely be an open label extension.
Can you explain what that is?
So when someone completes a study, and what we're describing here is, well, there's a dose titration, there's a follow-up. Traditionally, that was it. That was kind of the end of the study as a pharmaceutical drug developer. But if you were to involve your loved one in a clinical trial, and at the end of the study, you're feeling, he did well, or she did really well, and I want to continue it. There used to be no mechanism to do that. Most pharmaceutical companies are now offering what's referred to as an open label extension, which would mean doesn't matter whether you may have been on placebo or were on 352, you would be offered the ability of continuing into an open label extension, the same type of study design, and then continue with that treatment, typically for about a year as the pharma company is working on submissions to the FDA from a regulatory standpoint.
Thank you. Thank you. Yeah, I appreciate that. Sometimes our whole family doesn't know, and so I appreciate you explaining that. Any other questions? Great. Well, thank you. Let's give him a hand. Thank you. That was very helpful.